Hi, my name is Alina, and I'm going to be talking about the phagocytic response to focused ultrasound-mediated blood-brain barrier opening. So, as a little background, let me explain the phagocytes and their response to stimulus. First, phagocytes are commonly split into microglia and macrophages, which exist in the central and peripheral nervous systems, respectively. However, for the purpose of this talk, I'm going to refer to them under the joint group of phagocytes, defined as cells that express CD45 and CD11b. Phagocytes get their name as they are capable of performing phagocytosis, cellular digestion of pathogens. When exposed to stimulus, phagocytes perform a series of actions. First, within 24 hours of the stimulus, they send out signals to other cells to stimulate their migration to the area. Then, between 24 and 72 hours after stimulation, they send out signals to cause proliferation, the generation of new cells. After 72 hours, the cells that have migrated to the area gradually move away due to signals from other stimuli. Throughout the entire timeline of response to stimuli, phagocytes can convert from their surveying state into their amoeboid state and perform phagocytosis of pathogens in the area. This conversion is marked by the expression of protein CD68. Prior to this study, the Kline-Schoder lab has performed a preliminary assessment of phagocytes after BBB opening. These studies have found that phagocytes tend to express their amoeboid form after FUS-BBB opening, and that the interaction between the pathology in transgenic Alzheimer's mice and the phagocytes increases, suggesting significant changes after FUS-BBB opening. The objectives of this study were to 1. Quantify active phagocytosis through expression of CD68 after FUS-BBB opening, and 2. Quantify phagocyte migration and proliferation by looking at the phagocyte cell count after FUS-BBB opening. Briefly, the study methods are shown in this slide. Mice were sonicated bilaterally at the hippocampus at time of 0. Then an MRI was performed in order to assess the BBB opening. Then mice were sacrificed at 3 different time points, 24, 72, and 120 hours after sonication. After sacrifice, the hippocampus of the mouse was dissected and processed so that it was ready for flow cytometry analysis. The flow cytometry analysis started with a selection of the cells out of the debris. Then the alive cells were selected. Next, the count of phagocytes was developed by looking at the expression of CD45 and CD11b of the cells. And finally, of these phagocytes, the expression of CD68 was looked at in a histogram version, as shown on the bottom right. The phagocyte expression of CD68, 24, 72, and 120 hours after sonication is shown in the graph for 3 groups: those treated with microbubbles alone, those treated with focused ultrasound alone, and those treated with FUS and microbubbles, which means a BBB opening. After 24 hours, the FUS and microbubble cells have a strong increase in their CD68 expression as compared to the other groups. After 72 hours, this expression tends back towards the same point as the microbubble and FUS only, and then after 120 hours, there is nearly no difference between the 3 groups. This suggests that the phagocyte expression of CD68 in the FUS and microbubble group increases 24 hours post-sonication and reverses to baseline within 120 hours. Phagocyte cell count as a function of sacrifice time is shown for all 3 groups: microbubbles alone, FUS alone, and FUS and microbubbles together, as well as control. After 24 hours, there is no difference between the 3 groups, nor between the 3 groups and the control. However, at 72 and 120 hours, there is an increase in the FUS and microbubble treated cells as compared to the other groups. If you look at the phagocyte cell count as compared to the cumulative cavitation dose, there is a linear correlation at both 72 and 120 hours post-sonication. As expected, this same correlation is not shown at 24 hours. This correlation is stronger at 122 than 72 hours. There are 3 major conclusions of the study. The first is that active phagocytosis, as suggested by CD68 expression, increases 24 hours after treatment and reverses by 120 hours after treatment. The second is that the phagocyte cell count increases between 72 and 120 hours post-treatment, suggesting that the culprit is proliferation as opposed to migration of cells. Thirdly, the cavitation is correlated with the phagocyte cell count, 72 and 120 hours post-treatment. Future work will confirm that the increase in phagocyte count is due to proliferation as opposed to migration, and will look at the phagocytic phenotype changes after sonication. Thank you for your attention. as well as our funding sources. And I want to thank the Focused Ultrasound Foundation, as well as our other funding sources and my lab. Thanks for your attention.